This is part 2 of 2 on chapter 12, and we're looking at the elements of chemotherapy. So in the first part of this chapter, we looked at defining chemotherapy and then a few of the different types of drugs and a little bit of their origin. So this part, we're going to be looking at drug resistance and then trying to decide what type of drug you should be using to treat your patient. So the acquisition of drug resistance. So in the last chapter, I talked about MRSA, which is a type of Staphylococcus aureus that is resistant to a group of drugs. So that drug no longer works against the strain of bacteria. So drug resistance is becoming a bigger and bigger problem. So this is an adaptive response in which microorganisms begin to tolerate an amount of drug that would ordinarily be inhibitory or that would kill that microorganism. So basically, these bacteria, they are adapting to their environment. They're evolving over time to become resistant to these drugs. And this happens because bacteria have a lot of genetic versatility and variation in them. And this can either be intrinsic or acquired drug resistance. So it could either be like a mutation that shows up, or they can acquire drug resistance from other types of bacteria. So acquisition of drug resistance, acquired resistance, it can be spontaneous, like a mutation in some gene, and then this gene could cause the bacteria to become resistant to a drug, so the drug will no longer work. Or it can be acquisition of new genes or sets of genes via transfer from another species. And this can be done through the 3 different types of genetic variability that bacteria can do. So if you remember conjugation, where bacteria's cells can swap genetic information. It can be transformation, where bacteria cell picks up free DNA from the environment, or it can be transduction, where this DNA is coming into the cell through a virus. So those are the 3 different ways we can acquire new genes or sets of genes that can have this drug resistance on them. So different mechanisms of drug resistance. So these are ways that bacteria cells can actually shut down a drug, prevent it from working. So the first one up at the top, drug inactivation. You could have active penicillin that you're trying to use to treat an infection. That infectious organism could create an enzyme called penicillase that will inactivate the penicillin. So some bacteria cells can do drug inactivation. They can inactivate a drug, basically make it so it can no longer work. Decreased permeability, this is a second type in the middle. This is where your cell could have receptors on the outside that would prevent the drug from actually getting into the cell. You can have activation drug pumps. This is where the drug gets into the cell, but the cell has a pump that'll pump the drug back out. So basically, you keep on pumping the drug out before it can actually work against that bacteria cell. Number 4, you can change the drug binding site. So if the drug gets into the cell, but it has nowhere to bind for it to work, then the drug will no longer work against that cell. So here, for example, we have a drug, the little green triangle, binding onto a ribosome, and that causes the ribosome to become inactive. We could change that active site, so instead of its triangle, we now need a circle drug to fit in there. So that drug can no longer get to where it's supposed to get, it no longer can work. Or some cells, number 5, they can create alternative metabolic pathways. So if a drug is supposed to shut down a metabolic pathway, the cell can just form an alternate route to end up with the same product for that. So these are different ways that cells can become resistant to different drugs. So natural selection, which is one mechanism for evolution, natural selection goes along with drug resistance. So if you look on the left-hand side, we have a bunch of colonies. There's non-drug resistant cells, which are the darker cells, and then a few of the drug resistant cells. So you have a little bit of variation in your population. You expose those cells to the drug. The drug kills off the non-drug resistant cells, so we're killing off the darker cells. This allows those lighter colored cells to survive, and they're going to reproduce. And then you continue to expose your population to the drug, so all the darker colonies are killed off by the drug, and the lighter colonies survive because they have the drug resistance. So over time, this is just showing natural selection that populations will change over time, and they'll adapt to their environment. So some of these drugs, we talked about how we want to give humans and animals drugs that have a low toxicity. So we want the drug to work, but we don't want the drug to harm the host for our infection. But it's estimated that about 5% of all people taking antimicrobial drugs will experience some type of side effect. And some of these side effects can be very major. So you can actually have direct damage to the tissue due to that drug being toxic to the host. Other major side effects include allergic reactions. So for example, I'm allergic to penicillin. So I don't want to take penicillin for any infections that I have. Another major side effect is a disruption in the balance of the normal flora. So we all have normal flora, normal bacteria on our skin, in our mouth, in our intestines. If you disrupt your normal flora, you could end up having a super infection. And this is a major side effect. A lot of times antibiotics will cause stomach pain and diarrhea. That's a type of super infection. So basically you've killed off all your normal flora, so your body is no longer able to function as it normally would due to the super infection or missing all of your normal flora. So here, for example, is a super infection. So this person on the left, they have an infection in their throat, so they have a sore throat. So in their intestine, they have normal flora, which are the pink triangles, and the potential pathogen, which are the blue squares. So this person has an infection in their throat. They take a drug for the infection in their throat, but that drug circulates through their whole body. So the drug takes care of their sore throat, but it also kills off some of the normal flora in the intestines. So some of these pink triangles are killed off. This allows those blue squares to take over. And if those blue squares, if you get lots of them, it causes an infection, so it becomes a pathogen. And so now you have a super infection in your intestines, right here. So this is kind of how a lot of drugs, a lot of side effects we hear about are these super infections that will cause people to have diarrhea or stomach pain because one type of bacteria has taken over and their normal flora has been killed off by the drug. So some considerations when you're selecting an antimicrobial drug for your patient, whether if it's a human or an animal. First off, you want to identify the microorganism causing the infection. This will help you pick the right kind of drug if you know what kind of microorganism is causing the problem. If it's a bacteria, you can pick an antibacterial drug. If it's a fungus, you'll pick an antifungal drug, for example. And then you want to test that microorganism's susceptibility or sensitivity to various drugs. This is so that you can pick out the best drug that will work against your microorganism. And you also want to take into consideration the overall medical condition of the patient, if they have any allergies, or if they are taking other types of drugs that will cause the two drugs to interfere with each other. So you want to be really aware of your patient and what's going on in their body. So one, you want to identify the agent. So identification of your infectious agents should be attempted as soon as possible. So this is where you will take a specimen from your patient and send it down to the lab, or you can do a gram stain on it. So the specimens have to be taken before antimicrobials are given, so you know exactly what you're trying to deal with in this infection. Usually when you take a sample from your patient, you send it down to the lab, they're going to do a gram stain as the very first thing that they do. And then they can also do other biochemical tests to kind of identify more specifically what might be causing that infection. if it's like a streptococcus bacteria or a staphylococcus bacteria, so what type of bacteria. Or like E. coli or salmonella, for example. Then you want to test for susceptibility. So you can do the type of test we did in lab. It was called the Kirby-Bauer test. This is where we took the bacteria, we smeared it all over the plate, and then we dropped on little discs of different antibiotics on it. And then we measured that zone of inhibition and then compare that to a chart to figure out which antibiotic worked the best against that bacteria species. Another type is called an E-test, diffusion test. It's basically the same. Instead of putting the little discs down, you put a strip down, and on that strip it has very high concentration at the top and then lower and lower concentration going towards the bottom. So it basically tells you what concentration would work to kill that infection. So for example, the E-test strip on the left-hand side, you would probably use a concentration of 1 or 1.5, because that's the lowest concentration where we still see growth inhibited. The E-test strip on the right-hand side, again, you'd probably use about a 2 or 1.5, because that's the lowest concentration where we still see growth being inhibited. Any concentration less than that, you see growth that go right up to the strip. And then another type is called a minimum inhibitory concentration, or MIC. This is where you take your bacteria and you put different concentrations of your antibiotic into the tubes, and then you plate them, and basically you just look and see where no growth occurs, and then you want to use that concentration for your antibiotic. The MIC is what we just looked at. You also want to look at what's called the therapeutic index. So in vitro activity of a drug, so how a drug acts in the lab in a controlled environment, it's not always how a drug will work in vivo in the actual patient. So you want to also be aware, so the drug that works the best in these different susceptibility testings may still not be the best drug for that person. So you want to choose the best drug that has the highest level of selectivity. So you want to choose a drug that will work on that infection specifically to cut down on side effects. But you also want a drug that has the lowest level of toxicity that will cause harm to your patient. So you don't want to give a drug that would work except your patient's allergic to it. It's probably not a good idea. So you can actually, it's measured by something called the therapeutic index. So you don't want to make your patient feel worse than they already feel if you're able to do that. So you always want to take into effect patient factors. So you want to look for pre-existing medical conditions, existing medication that they take that might cause interactions, allergies. You want to look at the age, the gender of the person, and anything else that may you may want to take into consideration when treating your patient. So this is what nurses do, right? So they really have to know their patients, and they have to help them get the best treatment for their specific age and gender and existing condition.